Yeah, so today we wanted to talk about what can happen when the pesticides have reached their target. This could be the surface of plants or the surface of insects. Then, after that, we wanted to talk about bioassays and the field evaluation of pesticides. Depending on whether we have the time, we may move on to talk about pesticide registration and regulations, because these ones depend on bioassays and the field evaluations. They are more or less related. Today, I will try to present a full screen. If it's not clear, just give me a shout. Can you see the screen?
Yes, Doc.
What about now? Hello?
Yes,
Hello?
Yeah, I can
Can you see the screen?
it's visible, Doc.
see it.
You can see it?
I can see the PowerPoint slide, Doc.
Okay. Now I have moved it. Can you see the second slide? Are you seeing the second slide? Hello?
I'm fine.
Can you see the second slide?
No, Doc. I can only see the title slide. I can't see the second one.
You cannot see the second one.
Yes, I can only see the title slide. I can't see the second one.
All right.
No, it's still on the first one. I'm seeing the first one, target services for pesticide application.
Okay, so let me change. Right. Yeah,
No, Doc. It's not
it's
visible.
okay. So today we are going to talk about the target surfaces for pesticide application. So the targets for pesticide droplets are plant surfaces and the insect surfaces. We'll just focus on those two. Hello?
Hello, Doc.
Hello? Hello? Hello, can you hear me? Hello? Is Isaac in?
Yes.
Or is having some problems?
I can hear you.
Isaac? Hello?
Hello, Doc.
Is Isaac in? Okay, now I can start. Yes,
Yes.
let me start. So today we are going to talk about target services for pesticide application. The targets for pesticide droplets are plant surfaces and the insect surfaces. Now, if we start with the plants are covered by a barrier which prevents pesticide penetration. The outside layer of plants are covered by what we call the cuticle. And this cuticle has got several components or parts. So here I've got a diagram showing the cuticle. At the end here, or outer layer, we have got the epicuticular wax. It's a wax. Then inside it, we have got the embedded wax. And this area here containing the wax is known as the cutiny. So you have the cutiny, which is carrying a wax, and also wax is also found on the surface. Again, within cutiny, we have got pectin strands. Then after cutiny, we have got cellulose. Then we have got plasma membrane. Let's suppose we apply a pesticide. It has to pass through this cuticular layer so that it can reach the cells where it can disturb. For your information, some of the pesticides will end at the cuticle. They are not going to pass through it. Then we will have a selective killing. Yes. In the morning. Right. Sorry about that. So I was saying that the cuticle can prevent the pesticide from passing through. Here again, I've got the diagram, just like the first one, showing the cuticle. At the outer surface, we've got some wax. And this wax is also embedded in cutiny. And after cutiny, we've got pectin and the cellulose. And these are next to the epidermal cells. It's really the same diagrams. And the pesticide has to pass through this thick barrier to reach the living cells. Here we can demonstrate the entry of a pesticide into the plant system. The droplet can land on the wax, and it has to pass through the cuticle. Then it reaches the cells. And later on, it can go into the symplastic route or phloem route, which are the transport systems of the plants. But it can be very difficult to cross this cuticle. So the cuticle is composed of, number one, cutin, which is the major proportion. It is non-polar, which means it's not charged. It doesn't have charged molecules. It forms a sponge like a lattice. It's also a covering. It is a cross-linked polymer of various hydroxy and carboxylic acids, insoluble in most solvents. So for a pesticide to pass through cutin, it has to dissolve this cutin, which is quite problematic. Then cuticular wax, or just wax. This is composed of straight chain alkanes, or alkanes carrying hydroxyl or carboxyl groups. The hydrocarbons contain an odd number of carbon atoms, 21 to 35 altogether. Here we're just talking about the chemistry of the wax. It can also contain acids, alcohols, which contain even numbers, 20 to 24. So the outermost layer of cuticle is wax. Here I've got the structures of wax. So these are carbon chain atoms, which can be 27 carbons in one molecule, which is an alkane. Or you can have 26 carbon atoms attached to an alcohol, hexacosanol, for example. Or you can have an acid at the end, which is carbon 16 carbon. You have got 26 carbon atoms, but carrying an acid. Or you can have again 26 carbon atoms carrying this aldehyde. So there are various forms of the wax. This is just the chemistry of the wax. Now, within this structure, we have got what are known as stomata trichomes. Stomata trichomes are hairs and hydatodes, channels. These are just passages within the cuticle. Now, as for the hydatodes, they can only allow water out of the plant. They cannot be used as an entry point by the pesticides. You also have some hairs on the leaf which could trap pesticides. The stomata also do not form part of the entry of the points for aqueous pesticides. However, pesticides may enter via through the sensitive guard cells. They may enter through those guard cells. So the characteristic of the cuticle is that it is hydrophilic. That is, it can allow water to mix with it, and also hydrophobic. It may not mix with the water. So you may have the hydrophobic part on the outer layers and the hydrophilic part towards inside the plant. So when you are traversing the cuticle, you start with the hydrophobic regions, then you end up in the hydrophilic areas. If you are putting water, it means at first it's difficult for water to pass through the cuticle, but as you go deeper, water can easily pass through. Now, when the pesticides enter into the plants, the degree of pesticide penetration varies. We have what we call quasi-systemic. This is not, the pesticide cannot fully enter the plant. We have got the superficial deposits possessing an entry contact action. Now, systemic penetrates right through the cuticle and is translocated around the plant, unlike the quasi-systemic. Please note that lipoprotein membranes could prevent pesticide entry. So this was about the plants. Let us look at the insects. We have got the insect integument, which can form the barrier. You can see this at the scale. If you spray these insects, the droplets may not go through the insect. So basically, we also have a cuticle with the insect. We have a epicuticle, which is the outer layer. We have also the endocuticle. And we have some passages which are linked to sensitive areas. Yeah, probably this one is the symbol diagram which we may use concerning the cuticle. We have got the epicuticle right at the outer surface. Then this whole region is the cuticle. Then at the base, we have got the epidermis. But within this cuticle, we have got also passages which are used by some fluids to get out of the insect. So this barrier can prevent entry of the pesticides. Again, it's the same cuticle with the various versions of the cuticle parts. Again, another simplified version of the cuticle showing the various parts. So the cuticle is the outermost layer. The outermost layer of the cuticle of the insect is known as the epidermis. It separates the cuticle. So you have got the cuticle and the epidermis. Together, they are known as the integument. The cuticle covers the exterior part, and it can be rich in protein. Now, the inner layer has got glycoproteins. It also have other things known as chitin and tannin agents. But the epicuticle, that is the outer layer, it is rich in lipids and lipoprotein. So water will find it difficult to pass through the epicuticle. We have what is called the procuticle. This is secreted by the epidermal cells. This is 90% of the cuticle. What is referred as chitin is 60% water soluble polymer, which is N-acetyl glucosamine. So there are at least 3 kinds of chitin. The hydrophilic inner region of the endocuticle and the lipophilic outer exocuticle. So the lipophilic outer exocuticle may prevent water from passing, but the hydrophilic inner region may allow water to pass through. Well, the exocuticle contains melanin. Sclerotin is responsible for cuticular rigidity. It gives it the structure which is found in beetroots. There's also the presence of sensory cells in the exocuticular region. For example, the nervous system is the intimate target of many particles or pesticides. Waxes can prevent the uptake of the pesticides and may actually cause some selectivity. If an insect has got a lot of waxes, it may survive pesticide application. And the inner layer of the epicuticle is penetrated by waxy canals. So you've got wax which is embedded in this cuticle. So we can actually say that the cuticle is the first line of defense against the entry of pesticides and other chemicals known as xenobiotics into the plants and the insects. So the cuticle will serve a purpose of protecting the organism. So that is it. So what it means is that if we spray pesticides to the plants or insects, the first barrier will be the cuticle. And it can actually protect plants as well as insects. So it could be the differences in the cuticular structure which can determine whether an organism is going to die after pesticide application or not. So that's it on the pesticide targets, because the droplets first reach the cuticular surface, and then it may enter the cuticle or it may just stop there. So I will stop sharing. So this is it on the targets. We can see both the plants and the insects have got some similarities in terms of the cuticle, although there are some differences, but the function of the cuticle is to prevent chemicals to enter either the plant or the insect. Any questions on the cuticle?
No questions from me, Doc.
Okay.
None from me.
So I think we now know that nozzles can create droplets, and these droplets can be targeted to plants or insects. And we now know that the plants or insects are already protected by the cuticle. In fact, when we talk about formulations, there are certain additions which we can add to pesticides. Things like surfactants. We are trying to make the pesticide pass through the cuticle. So now I'm going to move on to bioassays. I will have to stop recording this